Hi Tanja, I'm very pleased to finally meet you. I've been recommended to talk to you by my doctor, who is looking after my daughter, who is on a spectrum of autism. He did some analysis using your lab, and I was really excited to come to talk to you because I really wanted to understand what was all those markers that came out after her testing. So could you please explain us a little bit why we use biomarker in the autism spectrum disorder?
Thank you very much, and thank you to have confidence in R.E.D labs. Indeed, the biomarkers in autism spectrum disorders are gaining more and more importance, and there are more and more evidence of their central role that they might play in the progression of patients on spectrum. As you know, autism spectrum disorders are complex. It's not one illness, it's not two, it's a spectrum, as it's in the name, which means that we have very different subgroups of patients with severe learning disabilities till extreme abilities in some areas, the patients with mild problems until the patients who have very severe problems and must be in special institutions. With respect to this, all these patients need a very specialized and personalized approach. We cannot propose the same approach to all these patients because their problems are not all the same. Now, the autism diagnosis progressed a lot in the last decade. Unfortunately, we understood that there is a huge progression of cases last 20-25 years. It's not only because the diagnostics are improved, it's because there are a lot of new cases. Now the debate why we have such a big number of cases that is rising every single year, it's a big debate, and we tend to know that it's a combination of different factors, as I will explain in a minute. But according to the very last information from CDC from US, in 2014, there is 1.7% of children aged 8 years in US that was diagnosed with autism spectrum disorders, which means 1 in 59. It's a huge amount. Now, again, it's a combination of genetic, environmental factors that impact on gastrointestinal tract, on immunity, and the impairment of gastrointestinal tract and immunity makes that the whole system is losing the performance, which opens the door to infections, then to opportunic infections, bacterial, viral, fungal, whatever, and then detoxification problems. Those small patients do not detoxificate as they should, and it's a sum of problems that are piling up and bringing more and more new problems, and at one point it's very difficult to propose something efficient to these patients. Now, the immune dysfunction is very present, and with different scientific publications, different groups, international, that are working on it, we can now make a kind of a small chart what could be at the least, at the minimum, potential causes, but no one can find the cause. There is not one cause there. It's a sum of different causes and conditions that bring the result of ISD spectrum. First of all, there is prenatal exposures, maternal and paternal toxins and pathogens. We know today that the mothers that suffered from infections or some health alteration during the pregnancy bring more children with autism spectrum disorders comparing to mothers that have no single problem during the pregnancy. It's not only infections, there are also dental amalgams, there are the diet, of course, we know today more and more about gut-brain axis with all the influence that microbiome maternal also has on children. Lifestyle factors, of course, smoking, alcohol consumption, etc. Gut dysbiosis, leaky gut in mother, etc. This is what we call prenatal exposure, plus prenatal exposure to microbes, which is a world apart. Then we have postnatal exposures, high sugar or carb diet, too much processed foods, too much toxins, non-organic food, GMO, nutritional deficiencies, too much pharmaceuticals, antibiotics, steroids, anti-reflux medication. A lot of children today are on reflux medication. Then, as I said, impairment of fundamental biological processes like detoxification, metabolism issues, then other lifestyle factors like stress. Stressed parents have a lot of influence on their children. They became stressed also. Outdoor exposures. Then we have environmental toxins. They're growing every day in our everyday life. And of course, there is genetic susceptibilities. And this is, let's say, one of the factors. Genetic susceptibility is what we call terrain, the susceptibility of children, because many children live in a toxic world that we have today with polluted water, polluted soil, polluted air, but they are not all on spectrum, which means that there is something that will make a difference, and this difference can make from genetic susceptibilities and impaired immune terrain at the beginning. And of course, more there are these factors, more severe will be disorders that one can diagnose. And that's why it's very important to adopt this biomedical approach, which is gaining more and more terrain with medical evidence and publications. Because first, autism diagnosis is suffering by the lack of specific biomarkers. It's not one biomarker saying, okay, like you can have serology testing for infectious disease. You can have, okay, you are sick or you're not sick. You do not have, and you will never have this in autism because there is not one biomarker. But we know today that several biochemical pathways are associated with ISD, and many testing panels worldwide in different laboratories and specialized centers are currently available for biomedical evaluation of multifactorial afflictions in general and ISD in particular. And with this, knowing that autism spectrum disorders are even more complex than most of multifactorial syndromes, needing thus a very specialized and personalized approach. And that's why we at R.E.D Labs, we did a big assessment of available literature. We are involved in many international groups aiming to increase the knowledge of biomarkers involved in autism. And we built our testing panel around three major topics that are immune disorders, intestinal dysfunctions, and infections. For all these three topics, these three major topics, there is a lot of possibility to investigate a lot of biomarkers, a lot of dysfunction that could be uncovered. Of course, we cannot test for everything at R.E.D Labs and worldwide. That's why we made a lot of collaborations in order to respond to different demands for even more specialized testing that are carried in very specialized laboratories like urine test for mycotoxins or heavy metal toxins, etc., etc. Now, as I said, immune dysfunctions in autism is very important part of biomedical approach, And if you read the paper published in 2010, almost 20 years ago, in Neurotherapeutics by Carrega and his colleagues, it says autism is a complex and clinically heterogeneous disorder with a spectrum of symptoms. Published findings have identified widespread changes in the immune systems of children with autism at both systemic and cellular levels. Together, these reports suggest that autism may in fact be a systemic disorder with connections to abnormal immune responses. Such immune system dysfunction may represent novel targets for treatment. And this is precisely what we like to spread as information. It's a spectrum disorder with a lot of involved factors that in first line physician has to go through to the whole history and to select the markers that are needed to be investigated for that particular child. We offer a very large testing panel to uncover different metabolic and immune dysfunction ranging from cytokine expression, oxidative stress, heavy metals, molds infections, macrophagic phagocytic activity to see if there is enough ability to clear the new infections, complement evaluation, then PG2 evaluation, which is a marker of inflammation, then ammonia accumulation, kynureninic and quinolonic acid accumulation, and many, many more. Immune response is very important because the immune response will be at the beginning of the problems. If the immune response is impaired, the downstream effects could be very, very dramatic. And we know that there is two types of immune response, cellular or called Th1, or humoral immune response, which is the ability to produce antibodies, which is called Th2. And those two needs to be always in balance. If you have a disbalance towards Th2, which is the case of many autistic children, then the body cannot face the new infections. While if you are in Th1, which means it's pro-inflammatory pathway pattern, then there will be no antibodies raised to the infection. That's why it's very important that the physician assess the balance between Th1 and Th2. When there is an infection, the body goes to Th1, but then when the infection is cleared, it should be back on balance between Th1 and Th2. And you have a very simple urine test, not even need to make a complicated blood draw, which within 3 minutes with color indication can indicate grossly if there is an imbalance towards Th1 or Th2. It could be a very easy starting point to evaluate immune disorders. Then there is assessment of natural killer cells, including CD57, and there is a lot of publications stating the lower number of natural killer cells in autistic children, both by Vojdani and his group, and we published also 2 years ago our own study on CD57 impairment in children with autism. After that, there is a quantification of prostaglandine E2 that has been found very significantly higher in autistic patients, recording an increase of over 91%. Knowing that PG2 is a key mediator of immunopathology in chronic infections, it shows very well how important this factor is to be assessed. And also, PG2 is the one who promotes Th2. And as I just said, Th2 will lock the patient in inability to face new infections. We recently published 2 months ago a report in order to emphasize the importance of assessing inflammation-related markers. It was published in Ames Molecular Science, and it clearly shows how many of inflammatory mediators are overexpressed in autism children, like IL-8, which is overexpressed in over 86%, another cytokine MCP over 50%, PG2 as I just said, prostaglandine E2, over 70%. Soluble CD14, that I will speak about later, over 83%. All these markers indicate that there is an inflammation. Besides these different immune markers, there is also a very, very, very big part of problems that can arise from oxidative stress, from heavy metals, from molds that will disable a good clearance of persisting infections. Then one should pay attention also on genotyping for methylation cycle polymorphism and also polymorphism of vitamin D receptor. These are investigated in different labs, in our lab also, and can refine the diagnostic of immune issues in autistic patients. As I just said, environmental exposure and oxidative stress are a big part of global biomedical evaluation. Oxidative stress is implicated in a large number of diseases, including neurodegenerative diseases and autoimmune diseases. And oxidative damage to cellular membranes can alter the permeability of the blood-brain barrier, and this can lead to some of cognitive symptoms observed in autistic patients. Intestinal dysfunctions are to be also considered as a part of the whole evaluation. Not everything can be explained by intestinal dysfunctions, but rare are the children with autism spectrum disorders that do not present any intestinal dysfunction symptom. Many publications are attesting that gut flora and gastrointestinal status in children with autism correlate with autism severity. The children with autism have much lower levels of bifidobacterium, less than 45%, slightly lower levels of enterococcus, and much higher levels of lactobacillus. Finigold and collaborators in 2010 published that the fecal flora of autistic children was statistically significant different from the fecal flora of healthy children. And for this reason, we propose to take an MSA test, which stands for metagenomic stool assay, in order to dig in detail intestinal flora at the bacterial level in assessing the percentage of good and bad bacteria and to see how at the best this can be corrected. The status of gut flora, some prebiotics, probiotics can be also prescribed. Another big problem is gastrointestinal inflammation, because several reports revealed a high prevalence of gastrointestinal symptoms, inflammation and dysfunction in children with autism, and mild to moderate degrees of inflammation are found in both the upper and lower intestinal tract. In children with autism, the presence of gastrointestinal dysfunction is often associated with increased irritability, tantrums, aggressive behavior, and sleep disturbances. We recently published, in the paper that I already cited, the importance of assessing intestinal dysfunction. And as you can see, we assessed soluble EGA marker, and over 68% of assessed children present abnormal soluble EGA. It can be either low or high. Almost half present low and high. Low soluble EGA indicates a lack of intestinal immunity, while high soluble EGA indicates intestinal inflammation. And with this simple marker that can be assessed in stool, this no invasive procedure for children, which is very important, one can already have a quite good information on what is going in the intestine of this small patient. A lot of publications now are done on gut bacteria and brain axis in autism, and I strongly recommend you to read at least one or two of them they're very, very, very comprehensively written, and you can more have an explanation how the gut microbiome and brain function are linked all together. You know, we are approaching another very important part, which is leaky gut. Now, some practitioners tend to say leaky gut equals leaky brain. Leaky gut hypotheses have been very nicely formulated by Dario Siniscalco and Nicola Antonucci in their paper from 2013, saying that autistic children show an elevated tight junction mediated intestinal permeability that allows the passage and absorption of dietary derived incompletely digested peptides at the intestinal lamina propria. Now, the leaky gut can be assessed both from the serum side, from the circulation level, which means that bacteria already crossed intestinal barrier, by testing the titers for IgM and IgG immunoglobulins in serum of patients, or it can be assessed through the stool by quantifying zonulin protein. As the zonulin levels rises, the seal between the intestines diminish. The zonulin is like a gatekeeper for the intestinal cells. It brings them tightly collated one to the other one, and if it's released to the intestine, it means that cells became more and more loose and the passage will become bigger and bigger, leaving the possibility to different molecules to go from intestine to the bloodstream. Also, bacteria are producing different metabolites, different compounds that can be very toxic for children and for their brains. For example, D-lactate is a product of bacterial metabolism, and it's neither produced nor metabolized by mammal cells. But typically, elevated D-lactate levels are due to bacterial infection or short bowel syndrome in humans. And due to slow metabolism and excretion, high D-lactate can cause acidosis and encephalopathy. The brain cannot distinguish between L-lactate that we are producing and using as a fuel and D-lactate, which is highly toxic. That's why we say it's one of the toxins for the brain. Another one very well known is ammonia that can be quantified in serum. Ammonia is derived from bacterial enzymatic action on ingested amino acids, and it's absorbed from gastrointestinal tract and delivered through the portal vein to the liver, which converts most of it into urea. And abnormally high levels of ammonia can result from colic or what we call enteric hyperammonemia, combination of increased bacterial production and increased gut permeability that occurs despite normal hepatic functions. And increased entry of ammonia to the brain is a primary cause of neurologic disorders, metabolic disorders, and some toxic encephalopathies. And that's why it's really important to fully understand this connection between gut microbiome and brain and neurocognitive impairment in autistic patients. Yet another interesting marker is soluble CD14, which is expressed in monocytes and macrophages and plays a critical role in the recognition of bacterial cell wall components like lipopolysaccharides. The extracellular part of soluble CD14 can be cleaved and released into circulation, into plasma or serum, where it will inactivate circulating LPS. And serum soluble CD14 are significantly elevated in patients with inflammatory bowel disease, Crohn's disease, but also in patients suffering from brucellosis and Lyme disease. And if you remember what I just said, bacteroides accounts for 30% of stool flora in controls and 51% in flora of stools of autistic children. This all together show you how important is the assessment of gastrointestinal issues in autistic patients. And we are coming now to the third big part of the biomedical assessment of autism patients, which is assessment for infections, bacterial, viral, and fungal. Most of the patients report an infectious onset of the disease, which means that the child was not presenting autism-related symptoms until such infection occurs, unless the child was born with autism, which is occurring in, according to the newest data, in 15 to 20% of cases. In the other cases, most frequently the diagnosis occurs between 18 and 36 months, and as I just said, following an infectious episode. The infection can be bacterial, and among bacterial infections, a very large part is on tick-borne infections. An association between Lyme disease and other tick-borne infections during fetal development and in infancy with autism, autism spectrum disorders, and autistic symptoms has been noted by numerous clinicians and parents. Different published papers give different statistics, saying that borreliosis may be a contributor for 20 to 30% of autism problems, while pathogenic mycoplasma may be a contributor with even 58% of cases. Kuhn and colleagues in 2012 suggested that long-term antibiotic therapy may be an effective treatment for children with comorbid with Lyme disease and autism spectrum disorders. When we say Lyme disease, we think about not only borreliosis, but about different tick-borne infections. Besides, Nicholson and colleagues in 2007 made a large study on autistic patients compared with healthy children, and reported that a large subset, 58% of patients, showed evidence of mycoplasma infection. And these results indicated a large subset of autism patients show evidence of bacterial and of viral infections. Several intra-monocyte pathogens have been evidenced in autistic patients. Several autism spectrum subgroups derive from intracellular or intra-monocyte pathogens, such as measles viruses, cytomegaloviruses, human herpes virus 6, and Yersinia enterocolitica. Human herpes virus 6 is a very important factor because it can be integrated in human genome and into chromosomes, and we can assess this because it's different if there is just an infection or if this virus managed to insert himself in chromosomes. Many opportunistic infections can follow the primary infections, and as I said at the beginning, on the impaired immune terrain where the infections cannot be cleared properly, new infections can arise very frequently, and the patients cannot cope with them. And this is a very fertile field for candidoses and different fungal infections. And precisely these fungal infections are very often disabling a good antiviral or antibacterial therapy to clear the infections. And that's why it is important to consider the fungal infection, especially if the patient is living in a moldy area, in water-damaged houses, etc., which can bring us to the chronic immune response syndrome depicted by Dr. Shoemaker, and, as I said, disable the successful treatment after that. A lot of targets can be evaluated when we speak about evaluation of infections in autistic patients. And it's the role, again, of the physician to see what are the pathogen on which to focus. A lot of publications have been done on pandas, on streptococcal infections, repetitive streptococcal infections in children with autism. All this is a part of initial anamnesis and the decision of the practitioner which pathogen are to be explored in a first phase. Because, of course, since that it's a sum of different problems, not everything will be solved with only one consultation or one set of testing. This is initial, what we call initial approach, when you have a selection of tests that have been documented in literature and practitioners know that can be linked with autism. But in second and third phase, some other pathogens can be considered because they can come as opportunistic and not as a part of primary set of infection. I hope that after listening to what was said, you can understand the need for biomedical assessment in approach of autism spectrum disorders. What is important to remember from all this is that more and more evidence points towards a combination of factors: genetic, infectious, environmental, immune, gastrointestinal, etc., being important in the development of chronic immune dysfunction, the cardinal finding in autistic patients. Unfortunately, in many countries, autism is still considered as psychiatric despite clear biomedical evidence. And for better management of this affliction suffering from lack of medical recognition, we offer specialty tests based on recent publications and extensive collaboration with medical doctors specialized in management of autism-related disorders. We developed dedicated testing focusing on global immune dysfunction, persistent or chronic infections, especially tick-borne infections, and intestinal dysfunction and intestinal inflammations. Of course, a wide network of new collaborations is needed for further advancement in autism biomarkers and deciphering and validation of new diagnostic and treatment procedures. Now, I would like to tell you a few words about who we are at R.E.D Labs. R.E.D Laboratories is a Belgian company developing tests for chronic immune diseases and intestinal dysfunctions. As I said at the beginning, we are involved in several international groups aiming to advance knowledge in biological markers of autism. We are continuously developing new tests according to the specific needs from healthcare providers. And all generated benefits are used for research and development of assay. We also have a very specific philosophy. We focus on setting up the tests that are not or rarely available elsewhere. But we also have a different collaboration with specialized labs in order to offer all the time larger and larger testing opportunities. Personalization testing panel leads to more efficient and rapid management of patients with complex clinical picture like the one we see in autism. We develop a lot of material in order to support our work and support understanding of biomarkers we are investigating. You can find them on our website. You can also mail us, you can call us, and you can meet us at different international and national conferences. And finally, I would like to suggest you some interesting lectures. You can find a lot of supportive videos, recordings, especially the recordings from Autism 1 conferences, where you can look for the presentations from Dr. Bradstreet, Dr. Ushman, Dr. Ruggiero, and Dr. Antonucci from different years. You can also find on YouTube a lot of presentations from Dr. Klingard. They're all very informative. I can complete the information I gave you today. Also, there are a lot of informations available on internet through the scientific papers and proceedings from conferences. Finally, for parents, I would strongly suggest you to join some parents groups who already went through this journey with their children and can exchange with you their experiences and to bring their own experience and to compare with yours. Please try to communicate and to discuss with your doctors, with your practitioners, healthcare providers, in order to discuss the possibility of biomedical assessment, because there are a lot of markers we can assess today and which can help them improving or adjusting the therapies they are currently prescribing. As I said at the beginning, different approaches and combination of different approaches is really recommended in order to complete at one part the diagnostic and on the other part the possibility of treatments that will evolve during the time, and like this you can really help your child. I hope you will find this information useful, and I thank you for your attention.